So first, I'd like to begin by acknowledging the traditional owners and custodians of the land on which we meet today, the Wurundjeri people of the Kulin Nation, and pay our respects to their elders past and present, and also pay our respects to the elders from other communities who may be here today. So it's a great pleasure to introduce today's speaker, Mark Lee, from my lab for his PhD oration. Mark joined my lab after doing an honours year in Dave Tarlington's lab, and I was really impressed from the outset of how hard he worked, and whether that was instilled in him in Dave T's lab or was something that came natural to Mark, I'm not sure. But for those of you who know Mark, know he's got a tremendous work ethic. More often than not, you'll find him working late in the lab, even at weekends and even on Christmas Day. And that kind of work ethic was hugely impressive. Although, just to point out to his partner Zoe, who's in the audience, that was against my instructions before you report me to HR. But Mark is really, as well as working hard, he takes pride in playing hard as well, in particular playing anybody who's played basketball with him, although his attempts to convince me that basketball is anything other than a futile exercise was unsuccessful. But it's great to have that ability to balance work and life, and Mark does that very well. He's really amassed a significant amount of data during his PhD, and some of that data he'll talk about today was recently published in PNAS. And we've also got enough data for at least another first author publication, which within the new restrictions of Melbourne University, hopefully we'll get to the point of writing that up before he submits, but we'll see. So with that, I'll just hand over to Mark and...
Thank you. Good day, everyone. I'm not quite sure the guy he described was me, because if you really know me and my Instagram, you know that I've been playing around a lot. So today I'm honored to give the opportunity to tell you the story about understanding the transformation of the killer protein BAK. So just to think about, to set the scene is, we think about death is a quite important part of life itself. And cells can die many ways, right? One way to go is via apoptosis. So apoptosis was coined by John Kerr and his colleagues almost half a century ago, where through their seminal observations under the microscope, they noticed one form of cell that's distinct from that of necrosis, where you form these membrane-bound apoptotic bodies. So they have this apoptosis models right here, where the dying cell will detach and fragmented into a membrane-bound bodies and then engulfed by neighboring cells. And therefore, they are immunological assailant. So through half a century of study, we know a great deal about apoptosis right now. We know, so on the right-hand side is elegantly summarized the apoptosis pathways by our honor student, Christine. So apoptosis can be initiated via two distinct pathways, either it's extrinsic or intrinsic, depends on where the death signal is come from. And the end game is always the same. You initiate the effector catabases, and the cells die via death by 1,000 cuts, coined by the Green's group. So today, my research focus is in the intrinsic apoptosis pathway, where the real story and the playground happens on the mitochondria. So why I focus that? Because it's quite important, and I believe the recent success of the anti-tumor therapeutic venetoclax has been proved this point. We'll take a look into this intrinsic apoptosis pathways. It's quite a simple pathway. So three family members govern this pathway. You have on the upper side the signal transducer messengers, so you call it. So the pro-apoptotic base-reonly proteins can be activated by that signal, such as DNA damage. And once activated, the pro-apoptotic base-reonly proteins can do two things, right? So one mode is to go directly activate the executioner BAK and BAKs, or they can function in a way that neutralize the pro-survival proteins and release the BAK and BAKs that's activated, bound to the pro-survivals, and leave them free to kill. So both BAK and BAKs, once activated, can form this dimeric structure, whereas they can then permealize the mitochondrial outer membrane termed MOP and release the protein content such as cytochrome C and enable the cell death. So we look closer to these family protein members, and what we notice is that all the BCL2 family members have this BCL2 homology domain. So the BH3 only proteins such as BID and BIM only contains one BH domains, whereas the pro-survival and pro-apoptotic BAK and BAK and the poorly characterized BAK contains multi-domains in there. If you look at the structural similarities, so what I took is the inactive BAK monomer and a BCL-XL. What we notice is that they are made of the 9 alpha helical bundles. They're globular-shaped proteins, and they look pretty much similar to me. The noticeable feature on those proteins is this surface hydrophobic groove, which colored in those red brackets, consists of the alpha helix 2 to 5. And this surface hydrophobic groove is critical for the binding of the BH3 only proteins. However, one thing we want to point out is the BH3 only proteins bind to BAK and BAKs is different to that to pro-survival BCL2s. By this, I mean is the BH3 only protein only transiently interacts with BAK and BAKs and then leaves BAK and BAKs to transform and carry out killer function. So in this schematics, I'm showing you what we know so far about BAK and BAK's transformation. So in a cell, that BAK is predominantly found in the cytosol, with its transmembrane tail tucked into the surface hydrophobic groove, drawn here. And BAK is predominantly found on the outer membrane on the mitochondria and association with VDAC2. So BAKs once translocate to the mitochondrial membrane, and since that point on, they behave pretty much the same. So BAK and BAKs can be activated by the BH3's only proteins, such as activated TBID, via the groove binding. And once activated, TBID goes away and leaving the BAK and BAKs to undergo a series of transformational change. So on this crude illustration, I want to draw your attention to two things that have been changed. That is the release or dissociation of N-terminal alpha-1 helix and exposure of this red bit, which is the BH3 domain of BAK or BAKs. The reason they have to do this is the exposure of the BH3 domain of the BAK and BAKs allows the dimerization, homodimerization of BAK and BAK. So one activated BAK or BAKs can insert its BH3 domain into the surface groove of another activated BAK and BAKs. From then on, this dimeric structure can then self-associate to form, presumably, this apoptotic pore large enough to allow the release of cytochrome C, and then therefore the cell death. So in this talk, what I want to present is our recent published work on PNAS, which focuses on first part is how does this BH3-only protein actually activate BAK at mitochondrial outer membrane. And then we want to focus on second part is how does this dimeric structure actually oligomerize to form this pore. So a quick illustration here. So how does this BH3-only protein interact with BAK at mitochondrial outer membrane? So we have these 3 cartoons here, and we look at the real structural information given. So think about from left to right is the motion image picture. And you have this inactive BAK. This is the surface group. It's colored red is this BH3 domain. And we know that via this MR structure, the BH3 peptide can bind to the groove. And then this is the end product, right? So I think it's pretty obvious that structural snapshots indicate there's significant conformational change happening along of the BAK activation. So we want to take a deeper look. So we know that, as I said before, that BID only transiently interacts with BAK in the model proposed as the kiss and run model. So, and we know that this groove, as I said, is later on required for the activated BAK to insert their own BH3 domains to dimerize. So which means this bit has to come in and vacate itself, make room for the dimerization. But along the way of BAK activation and those transformation things I mentioned, when BID actually runs away is unclear. On the right-hand side, we look at that motion picture, is after the kiss by BID, BAK also undergoes a whole range of different change. Sorry, the pointer wasn't working too well. So what we know from the previous study is the binding of the bit BH3 domain will expose partially of the BAK BH3 domain. However, the BAK full BH3 domain exposure right there happens only after the BAK alpha-1 dissociate from the rest of the protein. And we also notice this big change, like BAK exploding itself, that the core domain of BAK consists of helix of 2 to 5 and separates from the latch domain, which is alpha 6 to 8. So, however, we don't know the sequence of such change, so we don't know which one happens first, which one happens later. So the first question we need to address is we want to investigate the full-length protein interaction between the BH3-only protein, such as TBIT and BAK, at mitochondrial outer membrane. I know that similar kind of study has been performed before, but they are carried out in the truncated protein in the solution environment, which is suboptimal to understand a protein function at the membrane. So first part, the first problem we need to get around is Kisan-Run model, right? So illustrate on the left-hand side, that's what happens when BAK is activated by TBIT transiently. It goes on and oligomerize. So can we somehow, if we can lock BAK in its inactive form, does that stabilize BAK interaction so they remain kissed and not run away from each other? Then we turn our scope into the two endogenous cysteines in BAK. All right, so there are two endogenous cysteines in BAK. One is on N-terminal, which is the dashed line here, because we don't have a structure. As I said, the structure is truncated protein. The other cysteine is C166 on alpha 6 on there. And simply the first test we did is trying to see, okay, when we get the BAK on mitochondria, we induce intramolecular disulfide bound formation before activation, and we do it after activation to see what happens to those two endogenous cysteines. That's what happened. So draw your attention, so BAK itself should be running around 25 kilodalton, which is this band position. However, if we induce intramolecular disulfide formation, that means the protein has been constrained and somehow migrates faster on the gel. That's what we see here on this big blob. So that's the inactive form of fast migrating population of BAK. That's before activation. However, if you activate BAK with TBIT and then induce the disulfide bound formation, what you see is the loss of this blob, the inactive form BAK, and have active BAK and dimer, indicating those two cysteines might actually come apart after BAK activation. So obviously, the next thing we need to check is this required or relevant? So what we did is using a BMOE. This is an 8 Armstrong bivalent cysteine reactive crosslinker. So that means this can actually reach out to C14 and C166. So before BMOE crosslinking, if you like, if you add TBIT, wild-type BAK release cytochrome C from the pellet fraction to the supernatant, similar to that single cysteine controls. If you add the BMOE itself, it does not induce cytochrome C release, all right? And what happens if you have BMOE first and activates BAK? Single cysteine BAK mutants can still release cytochrome C, which means they're still functional. However, the wild-type BAK, once constrained, can no longer kill the cell. That to us, okay, this two cysteine come apart is required for BAK function. However, we know that cytochrome C release is on the very end of that motion picture I showed you. We want to check what happens in between. So what happens to the BAK? So the flow of experiment is where we take the BAK on isolated mitochondria, and we induce intramolecular disulfide titer of BAK, and then we activate with TBIT, and then we perform immunoprecipitation with BAK, with a conformation-specific antibody that recognizes the specific shape of activated BAK. And then we see, do we actually still get activated BAK anymore if we tether it? So here's the data. So in this experiment, same layout, the antibody we use is 4B5, which recognizes exposed BH3 domain of BAK. So in the BMOE tethered BAK, as shown before in the input, that BAK has been efficiently tethered and runs faster on the gel. And in comparing to the single cysteine controls, what we notice is that in the absence of cross-linking, you add in TBIT, BAK is activated, exposes BH3 domain, you get more BAK coming down with IP. And that happens still in the single cysteine control. And regardless of BMOE cross-linking, the single cysteine control still have more BAK coming down. However, in wild-type BAK, once you constrain the cysteine movement, you lost that increased BAK, suggesting that tethering endogenous cysteine inhibits BAK conformational change at early steps. So can we take this further? It's because now we know that we can lock back in a relative inactive form. Do we stabilize the beat-back interaction at all? So that's what happened. Whereas you perform the same experiment using QFE, so this QFE is the oxidizing agent inducing disulfide bond formation. So ideally, you think there's a much tighter constraint on BAK. So compared to single cysteine controls, whereas in the wild-type BAK, if you add in QFE, induced disulfide formation, do the IP of BAK, and beat comes down with it, which means if we lock the endogenous cysteine together, we inhibit BAK conformational change, and we are able to stabilize beat-back interaction. So that provides us a proof of concept that this is a doable approach to trap BAK-beat interaction on the mitochondria. Then what's next? So we want to lock back a different conformation stage. As I mentioned, that alpha-1 dissociation, BH3 only exposure, can we freeze back at different snapshots and see can we stabilize beat-back interaction with it? And luckily, through the previous work by our lab, Clark lab, and also structural biology lab, we have 3 mutants can do such things. The first one we use is the alpha-1,2 tether. So these 2 cysteines, if planted, can prevent that BAK BH3 exposure. The next one, alpha-1,6, similar to that endogenous cysteine place, you can prevent BAK alpha-1 dissociation. The next one, you can prevent the core latch domain separation, the one big explosion. And we ask the question, so if we induce this tether and then challenge BAK with BID, do the IP, do you stabilize BAK and BID? Obviously, if you think, if you can stabilize BID and BAK interaction in this setting, that means BID remains associated before this change happens. If BID is gone after you do the IP, that means this transformation change will likely happen downstream of BID dissociation and downstream to that the previous change of BAK. So here's the data. So that's the flow through. You have isolated mitochondria. You induce disulfide tether with QFE, and you challenge with BID, and immunoprecipitate all BAK from there. And ask, do you see BID-BAK interaction? So that's what happened. If you, using the alpha-1,2 tether, prevent BH3 exposure, you do see T-bit coming down with BAK, all right? Same thing with BAK alpha-1 dissociation, similar to that endogenous BAK cysteine I showed you. You have increased bit coming down with BAK IP. However, to our surprise, is where you prevent unlatching of BAK, there's no bit coming down with it. So this data, we interpreted a way that we can put an order of bit-induced BAK conformational changes, whereas the bit comes in to activate BAK in the groove, whereas we know the first tether can stabilize bit-BAK interaction, so we know that bit-BAK still remains interact at this stage. We know the alpha-1 dissociation, if you prevent it, you can have stabilized bit-BAK interaction, so they are upstream to that unlatching of BAK, because once you prevent unlatching, the bit is already gone before it. So that's all good. The next question we want to ask is, where is the trigger set on BAK? Is it only just this thing happen in the cell? Because if you want to design a small molecule to manipulate the fate or activity of BAK, you want to understand how this thing is activated, how many protein-protein interaction sites are there. So the question becomes, is there a bit-BAK interface elsewhere other than the BAK surface hydrophobic groove? So the first approach, when I started my PhD, we thought about, okay, let's use unbiased cross-linking of the full-length bit with BAK on isolated mitochondria, and then just use mass BAK to solve the cross-linking residues. Straightforward and really simple. Yeah, well, that's the experiment flow, where I trialed a different panel of photoreactive zero-length cross-linker conjugated with T-bit, put some UV light on, that diazorin should be really reactive, and they can then go on to BAK and hopefully give me some information about it. And I spent a year working on it, and I proved this approach is very difficult, because first, bit and BAK don't just stay there, they kiss and run, right? And the second part is the complex environment of mitochondria outer membrane. They have too many proteins and solute can be cross-linked there. So that was unfruitful. Then we turned back to a more targeted cross-linking approach using a cysteine chemistry where we're really good at. So still, I'm facing this problem that beat and BAK kiss and run, right? We then took advantages of this previous published mutant, the hypermorphic mutant, G94E of TBIT. This mutant, if you throw around to BAK, it does not induce cytochrome C release, but we don't know, no one knows more about it. We hypothesized that this mutant activity reduced is likely due to the reduced affinity for BAK, and therefore unable to induce those conformational change required for BAK activation. If you think about it, that's the catalytic reaction right there, right? If you have a wild-type bit, throw on BAK, the kids really good, and then go along to do their own things. However, this mutant, once you throw on, it still binds, but because of reduced affinity, it cannot kick off the BAK conformational change. So it might actually end up stuck there. And obviously, to use cysteine cross-linking chemistry, I need one cysteine on BAK, and the other cysteine has to be placed on BIT itself. Problem is, BIT itself, the P15 fragment, the active fragment TBIT, does not have a cysteine. We need to place a cysteine somewhere, and we chose the position IIT4, just behind the H0 position IIT3. And in case you wonder if that mutant is still active, we did a cytochrome C titration to show that IIT4C mutation does not really affect the activity of BIT. Compared to wild-type TBIT, you release cytochrome C really efficiently and similar dose. And now the question becomes simple. Can I isolate BAK on mitochondria, and then I first challenge it with the BIT mutant, let it become activated, and then throw in QF to induce that disulfide bound cross-linking. And then I immunoprecipitate it back and see, do I see BAK-BIT cross-link coming down with IP? So to lead you go through the data, because there's quite a few bands on there, so let's see what happens to wild-type BAK and cis-no BAK, all right here. So if you challenge it with wild-type TBIT, you see that, shown before, that QF induced that intramolecular tether of the unactivated BAK runs faster, and BAK forms dimer, similar to that IIT4C mutation alone that can actually activate BAK form a dimer. And IIT4C conjugated onto that hypermorphic variant G94E failed to induce dimerization of BAK. All the protein goes down, all right? Cis-no BAK doesn't have the many cysteines to play around, so it stays one lane. The easiest thing to observe the BIT-BAK interaction is actually on the BIT probe here, right? So BIT is 15 kilodalton and BAK is 25. You're expecting something around 40 kilodalton, so just above the 37 dot line. So here, there's nothing shown on the BIT probe. So we move on to try a panel of different cell lines with single cysteine mutations along the BAK and see what's happening. So this is the known structure of that NMR structure, where we see that there's the BIT B3 domain tucked into the groove, and that's H99 position on BAK. And that's I4C. Hopefully, that's close enough to induce a cross-linking. A single cysteine control, that's N124 on BAK, which is far, far away from this interface, should not give you a cross-linking band. If it does, then I'm screwed. Let's go along and see what happens. So here, the pattern is pretty distinct. In H99C, you have dimerization of BAK, and you have this lovely band right here. And if you look at the position, it runs the same size in the BIT probe. That's a band. Hopefully, there's a BIT-BAK cross-linking. And this band is not present in the N124C mutant. And I do acknowledge that there are other things cross-link happening due to whatever induced by QF. Okay? And to our surprise, that in the panel of BAK cell lines we tested in alpha-6 position and R156C, we observed the same cross-linking band happen and running on the same position here, indicating there possibly present a BIT-BAK interaction on the rear side. So if you like, behind this protein, there is a cross-linking happening. So I cannot convince anyone if I just throw in oxidizing reagent and happen to give you a bunch of bands. So I need to further test it. So we set up this competition assay. So how this assay works is pretty similar to the last one, instead of little changes. So the first line, that cross-linking band is what I just shown you in the previous page. And second and third line difference is I first incubate BAK with this cis-no G94E, which should still behave in a similar way after artificial cross-link performed, except that it cannot be cross-linked, all right? If I first incubate with this dot, if you like, and then incubate with the cross-linkable form, they should compete with each other if that's a genuine protein-protein interaction phase. And what you see is if I use the same concentration of two proteins, and you have reduced band intensity right there in both sides. And if I ramp up the adapt to 3 times more than the cross-link before, I essentially abolish that cross-linking band, suggesting, okay, I might actually got a genuine protein interaction. At this stage, all I can comment on is we're able to detect BAK interaction at groove as well as the alpha 6. However, I still need to answer the question, does this rear side matter at all? Is this all nothing but an artifact? So at that stage, we developed an independent approach to interrogate what's the functional relevance of it. So I set up this functional screen to interrogate important interface for BAK unbiasedly. So how this works is, here is your BAK on isolated mitochondria in a simple nutshell. And we utilize this obstructive label called pachymelamide. So BAK is 25 kilodalton, and PAK is 5 kilodalton. So 1/5 of the size is quite bulky. What you do is one end of it is the melamide group that reacts really well with cysteines, right? So you can label BAK on the mitochondria with that pachymelamide. And then I challenge it with TBIT and see the readout. So if that label interface is not important for BAK apoptotic function, you should still see cytochrome C release per normal. And if you see there's no cytochrome C release or impaired cytochrome C release, then I can further infer whether this interface is required for BAK interaction or required for the BAK self-dimerization or even the oligomerization interface from there on. So we did. And we take out to establish the assay. So here is, throughout the study, we only call residue is labeled when at least 50% of BAK on the gel observed is labeled with pachymelamide. And here's just a simple dose titration to establish condition. And obviously, I'm relying on the surface exposure of BAK residues to be labeled to actually generate a label. So here is a small, simple experiment, whereas in the wild-type BAK, I told you there are 2 cysteines, all right? And you see this 2 shift on the packet label, because I think that's it, the both cysteine got labeled. The other cysteine might got buried or potentially facing another protein. That's why it's buried, protected by packet label. Cis-null BAK doesn't get a label at all. Good. Single cysteine BAK, C14S, which is the C166 I introduced before, that is quite labeled on the structure and can get efficiently labeled on SDS page gel. M71, this residue is the first residue of the BAK B3 domain. As I said before, the BAK B3 is buried towards the core, and obviously you have a pretty retarded label on there. So now it goes through label a panel of the BAK cell lines I have. So this is a nutshell. I hope they can play. That in the spinning structure, you see all the blue dots are the labeled residues, right? And the black bits in the middle is the ones that resist label. So the labeling profile is largely consistent with the inactive BAK structure. So that is all good. Now, next thing is, put on the schematic. So these are the cell lines I generated and tested through packet labeling throughout the BAK. We stopped at BAK alpha 6, alpha 7 and 8, because these guys are already in the transmembrane domain. It will never get labeled anyway. So now, we see that all this pre-labeled BAK with PAK does not induce cytochrome C release in our studies. So that's very assuring. And what we found is, if you label YT9C, so which is right down in the BAK groove, you are able to inhibit cytochrome C release. So here, without pachymelamide, if you challenge that mitochondria with the 100 nanomolar recombinant full-length TBIT, you release full cytochrome C there, all right? And if you label with PAK, you see the labeling was pretty well. This is an over-bleached photo, I have to say. You protect that cytochrome C release fully, and all the cytochrome C remains in the pellet fraction, in the membrane. In contrast, for example, to alpha 2, the G72 mutant, all right? It gets really nicely labeled. However, all the cytochrome C is fully released. Then the question becomes simple. You just go along on the screen all the cell lines I have. And now the initial screen only reveals the importance of BAK group. So only YT9 is there. There's nothing on alpha 6. So at that time, I think, okay, great, life is dark. Why did I do this, right? This is a narrow field. I shouldn't do that. But also, when life is dark, you should let some light in there. So this guy popped along. So Michael was using my pack labeling approach to study the important functional phase of BAKs. And what he elegantly did is he used a titration of T-bit on recombinant BAKs. Then I thought, okay, maybe what happened here is because I'm using 100 nanomolar T-bit, that was too much. I'm overdosing the system. I'm covering the subtle differences. So can I lower down the dose of B to review that subtle change in the cytochrome C release? So first thing you do, obviously, is go to do a titration curve, right? So if I use 0.1 nanomolar of T-bit on the mitochondria, I have great release of cytochrome C already. And when I go to 1 nanomolar, you see the 1 nanomolar is already releasing as much as 100 nanomolar challenge. Then life becomes simpler. You need to take all these cell lines and rescreen them again with 1 nanomolar bit. And did it. And that's what happened. So that's YT9. If it blocks at 100 nanomolar T-bit, obviously it should block at 1 nanomolar. So that's good. And those 3 residues are for 6. So I think that's a day like I decided I'm giving up on this project, like just chuck it somewhere. And I found this result, that all 3 residues adjacent to each other on the same alpha-6 helix showed reduced cytochrome C release, whereas you see that reduction in the release fragment, and retention of cytochrome C in the pellet fragment. And I want to draw your attention that all 3 of them got a really good label on there. However, I cannot convince anyone by shades of gray. So I went along to put another cysteine there. So you put 2 cysteines in BAK, one at D16 and H164. What happens here is this mutant is still functional, indicated by the bit release, right? Bit-induced cytochrome C release, they're still functional. Really well labeled and 2 times label. And what you see is that dual label of paclitaxel alpha-6 resist at 5 to even 100 nanomolar T-bit activation. So here is great, okay. Now I know that this rear site is actually functionally relevant if I can block it with T-bit interaction. So what happened to BAK, right? What happened to its transformation that I stopped with paclitaxel? So I'm going to introduce you guys the gel shift assay in my lab that we use to infer the impaired BAK function. So this is not a Western blot. This is a blue native page gel, all right? So before you do anything to BAK, this is a seasonal BAK. If you take BAK and run on that gel, you see BAK distributed in 2 populations. One is this monomeric population. The other one is the VDAC2 associated complex form. And if you activate them with T-bit, you see the BAK become dimerized. This is dimerized BAK, and you lost the VDAC2 form as well. And if you pre-label with paclitaxel, then challenge this T-bit. Because it's seasonal, it shouldn't have any difference, right? Dimers runs here. And the antibody we use is the N-terminal specific antibody. To recall your memory that the N-terminal of BAK become exposed during its activation. So if it's activated, then the antibody can gel shift it. So before activation, obviously, BAK stays what it is. And all this activated dimer form can be efficiently gel shifted on the gel. You go to the next one, the single cysteine negative control, G4C on the N-terminus. Same thing before activation, 2 populations. Dimerized if you challenge with T-bit. The difference is want to draw attention is this population, right? The dimer with 2 pack labels, they run around here. And both of them can be efficiently gel shifted once you add in the antibody. That's good. Now look at what happened to the groove. So YT9 in the groove, that's where we can block BAK T-bit-induced cytochrome C release. Here, you still see dimerization. However, you don't see that dimer form anymore. You don't see that population. What you see is this single pack labeled BAK stuck in between the dimerized BAK and the monomeric BAK, suggesting BAK, once labeled in the group, failed to dimerize itself. And also, if you look at the gel shift profile, that the single labeled, pack labeled YT9 group seems to be retained somewhat in the gel shift assay, suggesting there also might be a problem with that early stage activation. And we turn our scope to what happens to alpha-6, because they also impair bit-induced cytochrome C release. Compared to the G4C negative control, you see that both the alpha-6 groups that they can still form that dimerized 2 times pack-labeled form of the population. However, there seems to be largely protected. You have a large population of the single pack labeled BAK retained there. And they give you this big smear, even if you store an antibody, suggesting there might be an impaired BAK activation there. And now, through the cross-linking profile, I can see an interaction. An independent functional readout, I can see functional relevance of both the surface hydrophobic group and BAK rear side. So now I need to give a reason. So why does BAK need to interact with TBIT at two sides, right? So why it needs to be kissed twice? I need a mechanism. So towards understanding of a mechanism of this two-side mode of BAK activation, we went to back and see, okay, this PAK labeling is actually pretty useful too to monitor protein conformational change. So a small experiment I did ages ago is where I see that monitor the BAK B3 domain exposure during activation. I've shown you guys before in the early step that the M71 is resistant to label before activation. Same here, right? So if before you activate BAK, you throw in the pacmaleimide, there's a really poorly labeled population there. And what happens is if you add in the pacmaleimide together with TBIT, you have this largely increased labeled form, which is consistent with the known transformation of BAK because it gets exposed during activation. So can I utilize that to observe more things? So I screened a whole bunch of other cell lines, and what you notice is that in this group, there's something interesting happening. So A130 is on the alpha-5, it's buried deep in the core of the protein, right? And what we observed is that before TBIT activation, if you pre-label A130 with pacmaleimide, there is almost like no label at all. And if you throw in the bit with PAK together, you see this drastic increase of label form. And another thing we do is if you throw in the pacmaleimide after you activate with TBIT, you still get increased label compared to the pre-label, but it's much less compared to during. So I think that is at least suggesting that BAK surface hydrophobic group become transiently opened and closed during activation. And does that make sense, though? We'll turn back to the known structure. So that's the data I just showed you. On the right-hand side, that's the the two-dose crystal structure, all right? So the inactive BAK, that's where A130 lies, right down and alpha-5 in the middle of the protein. If you look at surface exposure, you can't even see it. That makes sense. Pre-label it, it's barely labeled at all. Now look at the NMR structure where you have the B3 bound. That's where the A130 is. If you move the alpha helix away, you see the A130 become quite exposed in that groove. That is very consistent with what I saw on that SDH page shell. Okay, so now the question becomes, is this groove opening induced by bead binding like they said? Or is a prerequisite for bead binding? You have to have that groove open, then the bead B3 can bind to it. So I can't convince my lab head by just saying, so I have to do experiment. So we did that. So that's A130 itself, right? So before activation, you throw in pacmaleimide. It's poorly labeled. If you keep pacmaleimide in there and you're titrating in bed with increasing dose, you see the increased label of the BAK. Makes sense. It's consistent with last time. The last line is using mild heat to open up BAK. It's largely labeled. And what I did is put a cysteine on alpha-6 on the residues that are known previously blocks BAK activation. So R156. So I remember that I told you guys that R156 got really well labeled, all right? So before activation, all this one single labeled form is due to R156 rather than A130. There's no observable amount of double labeled form here. Now you're titrating, you label it, and then titrating in the TBIT. What happens is there's no induction of that second label form. So even though bit is there, PAK is there available, once you have the alpha-6 pre-labeled, the bit cannot interact with the rear side. There's no opening of the groove. You have no label there. And if you use heat to open up the protein, yeah, you get it. That result's being repixelated by another residue on alpha-6. So how we interpret it is that bit has to interact with that rear side of alpha-6 and try to facilitate the opening of that groove. Therefore, you get a label. Interesting enough is that rear side of BAKs. So Walensky's group ages ago reported that if they use a peptide that interacts with the rear side of BAKs, so this shallow group there, of containing the BAKs alpha 6, what it does is can allosterically open the BAKs groove, all right, and allowing the release of the BAKs tail, so BAKs can integrate themselves into the membrane. At that time, they argued they use a truncated form of BAK that say it doesn't happen on BAK because BAK is already there. You don't need it. However, I think my data and their data kind of suggest that that rear side is actually kind of a conserved mechanism for both BAK and BAKs, right? You need TBIT or other BH3-only proteins interact with it, opens the groove. In terms of BAK, you open up the groove so another bit can come in to activate it. And for BAKs, you can open up the groove where BAKs can first insert itself onto the membrane, and then that open groove is ready for the bead activation. So that's where we propose a refined model for BAK and BAKs activation that the BH3-only protein has to first come to the rear side, opens up the groove, and ready for the another BH3-only protein goes into groove that activates it and kicks off that sequential back change that I introduced before. So that's all good. We're coming down to the second part of talk. So we want to understand how these BAK dimers form this pool and to release cytochrome C, all right? So what do we know about it? So that's the dimer structure of BAK generated by Jason. If you think about it, the red side is the BH3 domain of BAK and inserted itself into form this dimer structure. Right-hand side, however, is not BAK, it's BAKs because it's a soluble protein. You can do it in solutions easier. So Ana Garcia generated this image using atomic force microscopy. Don't ask me in detail. I don't know how to do that. So, but you can appreciate that is how you imagine this thing as a Lego block can self-associate from this ring, okay? So you can think about the alpha 2 come together, glue it together, alpha 4, alpha 3, whatever. So this multimerization interface for BAK dimers to form that oligomer has long been held the holy grail of the field. If you think about it, is if all hope is lost, BAK is activated, form the dimer. If you can still impair or stop that dimer self-association, you can still save the cell, right? So there has been a number of reports on the potential protein-protein interface involves different BAK alpha helices, right? Essentially from 1 to 9, you name it. And even our own groups observed that the alpha 6 of BAK comes close together after activation. Well, since I'm already screened the entire BAK, why not I just screen her null again? So this time we use the mild heat, as I kind of hinted on before, that it's been found by Doug Green's group, that if you use 43-degree mild heat, you can simultaneously activate BAK. What this time you can do is comparing heat and bid. So heat provides a BH3-independent activation to push inactive BAK to active state, all right? And from then on, if my pacmylamide impaired or stopped cytochrome C release, the only thing I'm hitting on is the dimerization interface or the oligomerization interface. So we take on. So show you some examples. So in this set of screen, I rescreened it with 100 nanomolar TBIT and heat. So in wild-type BAK, obviously it doesn't matter, like you label or not. It fully released cytochrome C. In the groove, alpha 3, well, you block cytochrome C release, such as I shown before with 100 nanomolar TBIT. Alpha 6, however, if you label it, challenge with heat, it does not block cytochrome C release. And the drug memory, that data I shown before, that even you put 2 packs on the rear side, supposedly, you still cannot block heat-induced cytochrome C release. Therefore, that alpha 6 is unlikely to be a stable protein interface for the dimer to self-associate. And you go along to screen the whole BAK again. And that's the dimer structure. The blue bit is consistent is where all the label I put onto and try to stop cytochrome C release, all around this dimer unit, all right? Only 2 things we found to impair heat-induced cytochrome C release. One is that of YT9, which I shown before. The other is A130. So what happens? Is that an actual, multimerization interface or just block dimer formation? So went on, use blue native page, ask the question. So here is the signal BAK, simple, easy, like control. Monomeric BAK, BDAC2 BAK, and then you have dimerization happening along the activation line. Throwing PAK doesn't stop change anything. You have dimers. In the groove, Y89. So dimerize if you throw in bead or heat. If you throw in PAK, however, they're all stuck in between the dimers, right? You only have one labeled monomer. You don't have dimers, suggesting it's blocking dimerization. A130, however, is a bit naughty. In there, you have the dimerization per normal before PAK labeling. And once you have PAK label there, if you leave it unquenched, obviously, so the unlabeled form can still dimerize. The labeled form stuck there don't form dimers anymore. And using heat is you encourage that increase the PAK label as shown before on that SDS page gel, that all labeled A130 with PAK will fail to generate any dimer structures. So they're all stuck in dimers. So I think the take-home summary is the BAK activation-induced conformational change can destabilize bead-BAK interaction at the mitochondrial membrane. And we're able to tease out that the BAK core latch separation happens downstream of the alpha-1 dissociation based on when the T bead is coming off, right? Now we're able to identify a novel activation site that consists of alpha-6 that bead has to interact with both sides to activate BAK. And the reason for that is the beads interact with BAK rear side to facilitate the opening of the groove, and therefore you can have efficient activation. Doesn't move. Next one, the PAK labeling also suggests there's an absence of a stable protein-protein interface to form the BAK oligomers, all right? So if you think about chasing a pharmacological reagent to target protein-protein interaction to save your cells, that's probably unlikely to succeed. So our data kind of suggest and support the overwhelming trend of this apoptotic pore is either a protein-lipid pore at all, or if there's no pore, there might be the permealization is induced by the aggregation of random BAK dimer aggregates. So I did have a future direction, as Grant said, that because I don't have time to chase up that project, and my future direction due to that limit becomes essentially write up my thesis and hopefully get a job. So yeah, I want to thank everyone along the way. So first, obviously, is Grant himself. That's him before we met. Like, still really young, and Daniel Craig, if you like, that's what he called himself. He taught me a lot of things about how to approach this field, as I have zero knowledge about BCL2 family proteins, right? And also, most importantly, how to think a scientific question and to critically evaluate your data. And obviously, in this hit and run model, after I activate him, he becomes somewhat like a Bond villain. But the commonality, I think, is both of these guys look still happy somewhat towards me. And obviously, I need to thank my lab, the past and present members of the lab. They've been really supportive. I need to thank Ruth Clark and her lab members for teaching me all the reagents and details about BAK and BAKs. I also have to thank Michael, because without him, he's my angel. Otherwise, I won't be able to figure out anything. And obviously, I really appreciate along the way that the structural biologist has given me a lot of feedbacks that I can chase upon and try to figure out what's going on. And I want to thank my division, the Cell Signal Cell Desk, and also MGC, absolutely the best division in the institute. Questionable. I want to thank my PhD committee, and also I want to thank my friends, family, especially Zoe, to be being a really supportive partner along the way. And I'm open for questions. Thank you.
When are you leaving?
Tonight, it's my birthday.
Very romantic talk, lots of kissing. I'm sure there's going to be
Yeah, thank you.
Is BID the only BH3 only protein that can activate right by the rear site, or can some others do it as well?
This is not a planned question, all right? I thank you asked. All right, so we did something else. So there we go. So not only just BID. So rear site, you label it. I trialed with Beam and Puma. So one is the direct activator, supposedly. The other one is the indirect sensitizer. I have to say, these are not full-length Beam and Puma, because in the field we know it's really hard to generate them because they don't have a structure, right? I use a chimeric protein with the Beam and Puma BH3 domain. So if throw in there, at least I can say, okay, I can still block them or reduce the activation if I use their BH3. I don't have the full-length protein. That's so much I have on them. Yeah, and generated by calling, sorry. I didn't do that. Yes, Jerry.
Fascinating data, Mark. You may have said something about it, but I wondered, have you tried the possibility of using the peg blocking in more than one place, for example, Aptos 6 and the end of Aptos 5?
So dual label of different places. We tried it. We didn't try the exact place you mentioned. We're trying to do a bit more of that, but sometimes when you generate two systems on there, they're just not expressed. So I don't have the data, but I would like to try that on the list. Because one thing I think that didn't show is when I reduce the dose, I don't see more groove mutants become resistant. That's where we actually try the dual labeling, and the protein's not expressed.
The BAK VDAC2 that you label on your drugs comes up to 480 kilodalton. So both proteins are about 30. So what's the or what is the stoichiometry?
So we don't know. That's the question that I think our lab, Mark and Boris trying to answer. So what's that inhibitor complex? What are in there? The blue native gel markers don't reflect the true size of protein. It could be 4 or 5 protein in there which is not size, because you know the monomeric BAK is 25, right? It runs at 66. I don't know. He doesn't know as well. So.
It's likely got BDAC 1 and 3 in there, plus other components. We've done some mass spec on the complex, but as Mark said, you can take the blue native markers with a bit of a pinch of salt, because when you label monomeric BAK, it obviously shifts almost midway between the two complexes.
I thought that was a secret.
So Mark, in the Valencia study, they used a photoreactive peptide to identify the group as the binding site. Do you have any thoughts about why they didn't identify a rear site themselves?
Don't know. I mean, you mean the original Valinsky paper? Yeah. I don't know. They chopped the BAK into this truncated version that no one can reproduce it and throw in solution. Maybe it's a membrane thing. But also in the Cold Spring Hub conference, he and his group starting to use similar small compound and identify a whole bunch of rear site in all the BCL2 family proteins actually, not just the BAK and BAKs. He found something to MCL1 as well, so, which I think that's kind of indication that this actually exists in all BCL2s.
Are you aware of any BAK mutants in the Aptos that cannot be activated by BID?
Yeah, no, that's a short answer. No one in the field have those mutants. I want to. We tried to mutate all those 3, the arginine, the histidine into cysteine, so triple cysteine mutation. I test that in the cell lines. They are still active, comparable to wild type. I think is that serine mutation doesn't really mean much. I can be like looking at the wrong combination. I do have an idea from cancer patient samples. I mean, that's not reliable, but that D160, R156, and also H164 are highly mutated in patient samples. And I made those cell lines, I want to test them if I got time to see if they actually lost a function. They are mutated to D160Y, that's the one I remember. Anyone?
Okay, with that, just remains to thank Mark for a very